Hi, Assalamualaikum. My name is Azim shukri. Today I'm going to show you guys my poster about flow cytometry. Let's go. So today I'm going to show you guys one of the example of bio-optical signal which are flow cytometry. So what is flow cytometry? Let me show you guys the poster. So flow cytometry is an effective and sensitive technique that allows for the simultaneous collection of data on multiple biological activity, such as the expression of the surface indicator cytokines inside cells and communication protein of the cell cycle. Basically, flow cytometry is a technique that used to separate the cell, and we can know the color, the shape, and the types of the cell by using the optical radiation. So how the flow cytometry works? Here are the process. The first, we measure the single cells flowing through a detector system, and then flow cytometry can be performed on variety of tissue, and then the single cell suspension is then introduced into the flow cytometer into the cell-free buffer solution called the sheet fluid, which flow towards a laser aimed at the solution spot. And then the laser beams flow through the cell in the stream. The fluorescent dyes or antibodies affixed to the cell generate light at the particular wavelength when exposed to this laser. And then the captured information is examined to ascertain the size, the shape, granularity, and the presence or absence of particular chemical among the other properties of the cell. And then it can analysis help in identifying and sorting different type of cells in the sample. So here are the diagram of the flow cytometry. Like I said before, the the cell will be prepared and go into the shed fluid, and then we will add an antibody and fluid before we hit the laser. So the laser will hit the fluid and will be separate the cell according to the shape or the types of the cell. So here are the process of the flow cytometry. So flow cytometry has advantages and disadvantages. So the first one, flow cytometry is the most important technology for generating correlative information about the single cells. It allows the rapid and quantitative measurement up to the 20 parameter of cell, and its high specificity for discrete cell subsets and rare population. And then for the disadvantages, its highly multiplex data can also generate by genome-wide transcriptional profiling. However, this data does not allow for the distinction of signal on a single basis. And then various immune blotting techniques such as Western blot and polymerase chain reaction use cellular lysates and do not provide information about the single cells. And then in interpreting flow cytometry data can be complex, especially when dealing with the multiple parameter simultaneously. So besides that, here are the block diagram of the flow cytometry. Like before, we prepare the cell, and then we prepare the light source, and then it will go for the flow chamber where the cell will be added by antibody and fluid to be hit by the laser. And then it will go to the optical system which has laser that will separate the cells according to their color, and then it can detect by using light detector. So by the light detector, we can know the size, the color, the types of the cell. After that, we conclude all the data to the computer to be analyzed. So we back to the poster. So here are the limitation of the flow cytometry. So flow cytometry is limited when the requirement that analyze the cell is suspension. So sometimes it make the information of the tissue architecture and cell interaction is unavailable. Sometimes flow cytometry cells population with similar marker expression are difficult to differentiate and analyze because the cell is already complex and small. And then flow cytometry may generate a massive amount of data, making analyze more complicated. For overall, flow cytometry is a pivotal tool for the analysis of cell subset and their complex interplay in immunological and biological process. It has advanced our understanding of the immune system and likely to play a fundamental role in the future. So basically flow cytometry is right now is one of the important things in medicine to we know the type of cell and the we can disease cancer cells, and we can know how the cancer cell by using the light radiation. So here are the journal link and YouTube link. You guys can QR code it to find more information about flow cytometer. So that's all for me. Thank you so much for you guys because lending your ears. So we meet next time. Let's go. Bye bye. Assalamualaikum.